So we left off at learning objective 3, which is to describe the nature and significance of the blood-brain barrier. And so we're going to look at the structure of the blood-brain barrier and talk about why it's so important. So blood supply to the nervous system is extremely important. The nervous system, although it's about 3 to 4 pounds, the brain and spinal cord, this is going to receive a very large portion of our total blood supply, our total cardiac output. So about 15% of our total cardiac output, about 15% of all the blood that's leaving the heart will eventually go to the brain and spinal cord, although the brain and spinal cord are only about 2% of body weight. So that's really fascinating, right? The brain has a high metabolic rate. It's highly active. It's going to use about 20% of oxygen that we consume at rest and about 50% of the glucose that we consume at rest as well. So a lot of the brain functioning is going to be highly dependent on constant energy, which is received through blood supply, right? Oxygen, nutrients that are coming in through that blood supply. Neurons are aerobic in their cellular respiration, so they require oxygen. We talked about this idea that the brain really prefers glucose as its main source of oxygen, just because neurons need to break down this glucose with the presence of oxygen. So having a steady glucose supply and a steady oxygen supply is extremely important for the brain. There isn't a lot of glycogen stores. There's a small amount in the glia. We talked about astrocytes having this ability to provide some glycogen stores, but other than that, there's very little glycogen being stored in the nervous tissue, and fatty acids are not very well utilized in the nervous tissue as well. So it's extremely important for the brain to be able to get glucose from ready supply coming either from the liver or coming in from our diet. Ketones are going to be utilized under extreme conditions. So under very extreme conditions or circumstances, can the brain actually tap into using ketones? And ketones, by the way, are the breakdown products of fatty acids. So let's say you're on a ketotic diet where you're not taking in carbohydrates at an appropriate level. The body can switch over to oxidizing fats and then making ketones, which can supply most of our tissues fairly comfortably, but the brain is not going to be an ideal situation to use ketones. And so this is where we can have fatigue, cognitive fatigue, or maybe some cognitive decline if we have a situation of an extremely ketotic diet or extremely low carbohydrates or just not enough glucose. On that note, we can think about things like strokes. So strokes are a situation where there's decreased blood supply to the nervous system, specifically to the brain. And this can be an occlusive type of stroke, which is where a blood vessel is blocked. And that can be, there's a lot of plaque buildup, a lot of atherosclerosis in the blood vessels that can actually occlude the lumen. And then the other type of stroke is a hemorrhagic stroke, which is where the blood pressure is extremely high, more chronically, right? If you have a long-term chronic hypertension, then you're going to have weakening of the intimal layers of blood vessels, and that can lead to rupture of those blood vessels. So the two types of strokes we can see is a blockage of a blood vessel, an occlusive stroke, or a hemorrhagic stroke, which is where there's a tear or injury in the blood vessel. Wow. Let's talk about the blood-brain barrier. So the capillaries help to form the site of exchange between the blood and the interstitial fluid around the nervous structures. It's a thin layer of endothelial cells. These cells are joined together by tight junctions. And so there's some diffusion that can happen across this blood-brain barrier, but only for small molecules, small, nonpolar, oxygen, carbon dioxide can diffuse across, but larger molecules and pathogens and that type of thing will definitely be monitored by way of the blood-brain barrier. So it is the special anatomy that is limiting the amount of exchange between our blood vessels and capillaries, and then what's happening in the nervous tissue, the interstitial fluid around the nervous tissue. And this is formed by the astrocytes. So we looked at the structure of the astrocytes. So here's what they look like. And they have these star-like projections, right? These kind of projections from the central part of the cell. And the foot processes of these projections actually sit over the endothelial cells. So basically, they're sort of acting like a security checkpoint, determining what can leave the capillaries and make its way out into the interstitial fluid of the nervous system, and then what has to stay behind. So in this way, the astrocytes are really securing the junction between the blood and the brain. Additionally, there are tight junctions. So in between these endothelial cells of these capillaries, there are these tight junctions that are also preventing any paracellular transport or any leakage. So in a typical capillary, anywhere else in the body, notice that there are these tiny pores in between our endothelial cells. This can allow certain things to freely move in between the interstitial fluid and the plasma, which is inside the blood vessel. Whereas in the brain, that's not ideal, right? That's not the case. The tight junctions help to secure any pores and prevent that type of free movement. And then the astrocyte foot processes also add an additional layer of security, really monitoring what can leave the blood and make its way out into the brain. If we look at things that can go through, so typically we have transport-mediated or carrier-mediated transport of selective hydrophilic solutes that can be moved through the endothelial cells via these transporters. But there's not a lot of diffusion that can take place, right? Because these pores, as you can see in a typical cell, are not open. So in order to leave the capillary and go out into the brain, it's going to have to be a small hydrophobic solute, right? So small lipophilic or hydrophobic solutes like oxygen, carbon dioxide, those things can freely move. But other particles, proteins, glucose, those cannot be transported, those cannot be diffused across this membrane.